under environmental control conditions and working with molecular biology. So it's something that we need to do when we are biochemists, that we need to create knowledge in the lab and then try to apply that knowledge in the field. So that is what I did during my postdoc, apply all the knowledge that I get during my education into the field. So when I started my program, my PhD program, I was interested in the interaction that happened between plants, among plants and pathogens and vectors. And this interaction can be symbolized by this triangle, in which you can find different comparisons that you can analyze, or interaction that you can analyze. For example, how the vector interact with the pathogen. There are certain pathogens that give you traits that are beneficial or can be detrimental to insects. In the case of the Candidatus liberibacter solanacearum, or the Candidatus liberibacter solanaceae, they have certain traits that help the insect to survive or have a detrimental effect in the case of certain the haplotype LSLB that reduce the reproduction but gave them other traits. In the case, there is another interaction that happened that is the vector and the host. In this case, what we are trying to do in here is to determine which are the visual and the chemical cues that make the insects, make the plants more attractive to the insects. There is another one that is the pathogen and the host, is how the plant responds to a specific pathogens. In the case of how the citrus, I'm going to show how the citrus respond to silas in different time points. Also, there is another that is more complex, is when the vector is associated with the pathogen and interact with the host. There are other level of interaction in there. So as I say, I was a biochemistry. I was a biochemist, and I've been mostly working in molecular biology. And in this area, I've been working with different omics. I have been working with genomics, in which we sequence the DNA, and we are soon we are going to publish the genome of Bacteria querelli. We have been doing genetic profiling, genetic mapping, and I had used recombinant DNA technology to create or to do some genetically modified organism, specifically in Drosophila in Chile. I have been always working with transcriptomic, and in there we analyze we do the RNA sequencing, we determine the profile, the expression profile, and with the final goal is to determine how the transcription is being regulated. But it's not only that you have the genes expressed, transcribed, you also need to study what happened post in the translation. In this case, I've been studying proteomics, in which we identify the protein. In my case, I use MALDI-TOF to identify proteins, and we quantify those proteins, some using Western blot, and also some proteins are need some post-translational modification. For example, some enzymes need to be activated. They are pro-proteins, and then they change to the pro-enzyme, and then they change to enzyme to have certain activities, and some of them are synthesize metabolites that are important for the plant, metabolites or hormones. So in this case, I've been using metabolomics, in which we profile those metabolites, we study some intermediates, and also the hormone and signaling molecules. But not everything is molecular biology. We need to transfer that information to the field, or to use that information into the field, or to understand how the organism interact with the environment. In my case, I've been interested in integrated pest management, that is IPM. In Florida, we use mostly of the focus of the IPM in citrus is related with the chemical control to reduce the population of Asian citrus psyllid. And in this case, we have using the conventional insecticide, but that has generated a problem in Florida in which the insects are now resistant to several insecticides. And to reduce that, to remove that stability, to remove that stability, we need to do insecticide rotation. In the case of the citrus and the Asian citruscillid, if you know about, a little bit about the biology of the Asian citruscillid, they only laid eggs on the new in the new tissue, specifically in bud breaks and also in fair flatch structures. And we create a model in which we are using the threshold and phenological models. So our insecticide applications are associated with the injury threshold and at the same time to the plant phenology. I use a little bit of the biological control. I did not have enough time I will not have enough time to touch that part. But in this case, we use parasitic wasp and predators. We are studying the diversity of them. And then we have the cultural control in which we can use windbreaks and use reflective mulch. So as I mentioned before, or as Chloe mentioned before, I was in Texas A&M working with Silet and Burindegi, in which we were focused on this disease that is several chip that happened on potatoes. And this is an image of what happened with the potatoes when they are infected with the pathogen Candidatus Libreactor solanacearum, which is transmitted by the vector Bacteroides kereli. At date, between 8 to 10 different LSO haplotypes have been identified, but only the haplotypes A, B, and F are associated with Bacteroides kereli. And within these haplotypes, you have different level of pathogenicity. For example, the most pathogenic haplotype of LSO is LSO-B. It kills the plants in between 3 to 4 weeks. In the case of LSO-A, the plants get infected and they get stunted, but the symptoms are lower. And this disease in Texas and in other states has cost millions of dollars in United States and in other countries. So when I started my PhD, they proposed three different projects. One of them was to study the immune response, specifically how is the interaction of LSO with the psyllid in terms of studying the apoptosis. But in my case, I was interested to how in another project that was how can we reduce the population of an insect? What do we need to study? So in that case, I was thinking like, oh, I want to study the reproduction. Okay, so now what we can do in the reproduction? In the reproduction in insects, there is an important process that is called vitellogenesis. That is when the vitellogenin is synthesized in the fat body cells and finally transported into the oocyte. And in there, the vitellogenin is called vitellin, and vitellin is the main nutrient of the embryo to finally develop. So in Bacteroides kereli, I did these three main things. I did the identification of the vitellogenin gene. We determined the expression and how is regulated, how the expression is regulated. So the first thing that I did, I took females of Bacteroides kereli, I extract the RNA, and we did race PCR, and I amplify a fragment or a DNA amplicon that was 6.6 kV, and I clone this the whole gene into a vector, into a plasmid, sorry. That is a PGMT. We clone it and we fully sequence this gene, and this transcript has an ORF of 6.3 kV. And then I compare the sequence to other vitellogenin genes that are previously described, and I perform this phylogenetic tree, and using Bayesian analysis, we our vitellogenin clade with other hemipterans. After I did that, we determined the expression. For this, previously I checked if vitellogenin was expressed in males, and it did not, and it was female specific. but I would like to know how this gene is being regulated in females. So we use females of different ages, 1 day, 3 days, 5 days, and 7 days. Also in different stages, virgin and mated. And we determine that the Vitellogenesis gene is upregulated by age and is upregulation is larger when the female is mated. Down here in the panel B is a SDS gel in which we extract the protein from 10 insects, and using 20 milligram of protein, microgram of protein, we analyze the abundance. We knew by informatics that this protein it was near to 150, 160 kilodalton. And then by visually, we found that one of these band was increased with age and also in different mating stage. And we sent those two bands for sequencing using MALDI-TOF, and we determined that those two bands correspond to vitellogenin in different conformations. Probably one of them is the one that is directly synthesized from the in the hemolymph, I'm sorry, in the fat body, and then move it to the then move to the oocyte, and this protein has different post-translational modification that may vary the size of this. So then in insects, if you know the insect, the regulation of the reproduction is mostly by two hormones. One of them are the juvenile hormone, and the other one is the exdysons. In the cases like you have several forms of juvenile hormone, 1, 2, 3, and then you have the active form of exdyson is 20-hydroxyexdyson. But in hemipterans, one of the the juvenile hormone is the one that regulates the process, the biological process. So I exposed topically, or I applied topically juvenile hormone 3 to the insects. I analyzed the expression after 24 hours, and we found that in two different doses of juvenile hormone, the expression was upregulated. Then in the same insects, after 96 hours, we collect those ones after post 96-hour post topical application. We collect these insects and we observe the abundance of the protein. And in here you can see how the amount of how the abundance of vitellogenin increase according with the dose use, the dose of the juvenile hormone. So we found that juvenile hormone is a main controller of vitellogenesis in Bacteria baccherelli. So in here, it's not only we did not only determine the expression of the gene and the abundance of the protein, we also dissect those females, and I analyze the number of mature oocyte. In here, I'm showing you this is a mature oocyte, this is the ovarioles, the oviducts, and in here there is a structure of the spermatica. Then, but in this case, I forget to mention, we use only virgin females. And when the when the spermatica has this shape, is an indicator of that the insect has not mate. And you can see this pretty easy and straightforward that as soon as we increase the doses of juvenile hormone, there is more mature oocyte. And this is exactly what we observe. So this is a small summary of what I did. So, but the reproduction is not only one gene. Vitellogenin is the final product of the reproduction. There are other factors or transcriptional factors that are involved. During that moment, we did not identify the main receptor of juvenile hormone, which is the metroperitonian receptor. We found another gene that is a Kruppel homolog 1, Uh, and this gene has been involved in vitellogenesis in other insect species. So we followed a similar experimental design. We identified the Kruger homolog 1, and we determined the expression. And finally, using artificial diet, we apply double-strand RNA and see what happened with the vitellogenesis. This is what we observe in the gene expression. There is a upregulation of the gene as soon as the insect age. Uh, we also at the same time we analyze the expression of vitellogenin in the same stages. B mean virgin and M is mated, and we found that the expression was kind of similar, and as it was related, it was we did an assumption that Kruger homolog 1 could be involved in vitellogenesis. So then we design an experiment in which we fed the insect for 24 hours with double-strand RNA to Krupel, and also to the control that we use EGFP. A group was without juvenile hormone, and a group of female were exposed topically to juvenile hormones. And in here we saw that we did not find statistical differences in the expression of Krupel without and with juvenile hormone, but in the case of that is for the control, and in the case of Krupel homolog 1, the expression of this gene was highly reduced. At the same time, in the same samples, we analyzed the expression of vitellogenin, and we observed for the control that post-application of juvenile hormone, it was a higher expression. But in the case of the females that were fed with the double strand, that activation was completely knocked down. So female fed with double strand RNA had a reduced expression of Krupel, and females were insensitive to the topical application of juvenile hormone compared with the control females. I follow almost the same protocol that I did for vitellogenin. We dissect those females, and what we observed that the females the control females had a higher amount of mature oocytes, and in the case of the mature oocytes on female fed with the double strand of Krupel, we did not find any mature oocyte, or it was really low. So with this information, we can impair the reproduction and finally reduce the psyllid population as soon as we apply this technology on the field, or we found a technique to apply this technology in the field. Maybe creating a trap crop system that can express a hairpin RNA against these transcription factors. Then after I finished my PhD, I moved to Florida, in which I was working with Dr. Lucas Stelinski in this disease that is called Wanglong bean or citrus greening. And in here you can see phenotypically how the fruits look when they are infected with HLV, or not infected with Candidatus liberector asiaticus. That is the causative agent of HLV. And this bacteria is transmitted by the Asian citrus psyllid or Diaphorina citri. Several haplotypes of Silas have been associated with the Diaphorina citri, but there are so many. I did an experiment in which I used different primers, and at least I detect like 10 different variants of 10 different haplotypes of Silas in one grove. And this disease has devastated the Florida citrus production. 20 years ago, Florida has almost 900 hectares or 900 acres, and now they only have 500, and it's reducing year by year. So then, like my point is like, okay, how this disease interact is really complex. When the trees are infected, sometimes you find the Silas by PCR, you detect the Silas. Another time you don't detect. So one of my objectives there was to know when the bacteria is in the mature leaves and when it's not, to probably create strategies later on to reduce the bacteria when it's in the aerial part of the plant. Another problem that we have, that the rate of infection of insects of ACP vary a lot during the year, going from 10% to sometimes 90% of infection. So it's it's really variable, and we need to spend a lot of resources to finally reduce the incidence of HLV. And this disease is across all the southern state that has citrus. So I did a similar study there. We know by literature that the Diaphorina citri has at least two different color morphotypes. Um, I analyzed the expression of them and the reproductive output. These morphotypes are blue, green, and gray and brown. And the morphotype, the blue-green, has been associated with high tolerance to insecticide. They can fly more than the gray, and also they have a greater reproductive output. They can lay more eggs per female than the gray female, but no one knows about what are the biological processes that allow this. So similar approach, we identify the vitellogenin gene. The Diaphorina citri has a transcriptome, and it was at least has 2 or 3 different vitellogenins. We use the one vitellogenin A1 that this was the most similar to our vitellogenin Bacteria querelli. I did a phylogenetic tree, but I don't need to show you. They clay together. As you can see my paper there, my manuscript, and we analyzed the expression of vitellogenin in different ages when the females were 1, 7, and 14 days old, and we found that the expression of vitellogenin was upregulated in green females compared with the grays. I dissect those ones to understand, okay, is just the expression or is something else? In here you can see how the ovarioles look when the females are 1 day old in gray and blue. And you can see how big they are after 7 days. The insect that is green has a higher metabolism. They develop faster than the gray ones. And until now, we really don't know why we have the gray and why we have the green. I did an experiment to analyze if it's something genetic. And after the generation 13, we still have gray females in a lower percentage. So I'm continuing this study here in which I'm crossing just green insects and then using the progeny to cross them again, and finally see if I can increase it. We did not see any change. We saw a lot of significant differences um, after the generation 10, in which the percentage of green insects increased. But what do we also observe, that the females tend to be green the whole life. The male change from green to gray, and then they die. So I'm trying to think exactly what is exactly is happening. One of my ideas is probably the color is associated with the sexual chromosome, but I'm still thinking about this part. So this is just another plot saying that the green and blue insect has higher number of eggs, and also they have higher number of mature oocytes. So this is what we found, and these two morphotypes, and this is the summary. High expression of vitellogenin in green females. They have an increased number of developing oocyte, and also they have a higher reproductive output. So as soon as they are developing the mature oocyte, they are laying the eggs. This was the part I did with the insects. And now one of the questions was addressing where there is a need for a continued vector management under HLV stress. Approximately like 95 to 100% of the citrus trees in central Florida are infected with psilas. So if we have that one, is there a need to spray the vector population? Because if the trees already have the bacteria, there is any reason for us to apply any pesticide. In that moment, we have two main hypotheses. One was called the superinfection, that is when the tree is exposed to multiple inoculation of the bacteria. So those trees are going to harbor a higher amount of, are going to have a higher titer of silas if superinfection occur. And the other one was superinfestation, in which the tree is exposed to higher number of insects. And are they going to develop symptoms that are similar to HLV? That was the question. In this case, there is a hypothesis that I'm gonna touch today, is like decreasing silas inoculation frequency will reduce the silas titer and improve the plant health. So for that, I did a field study and also a laboratory control environment study. In the field study, we evaluate the threshold and the phenological models and their association with HLV. The objective of this one was to evaluate, or technically was to compare the conventional grower practices versus the spray coordinated with flash phenology to suppress ACP in commercial growth. The specific objectives was to quantify the impact of the management protocols on ACP population and silas titer, and we examine the association between pesticide application, ACP population, and silas titer. To quantify the ACP population, this is a normal protocol that we had in the lab. We use 10 trees per block, 3 blocks per location. We divide the trees in section, like the left, the middle, and the right. And we tap those, just tapping and counting the number of insects. And this is what we found. For the tapping of insect, we did the experiment from this since March 18 to April 19. The grower practice are highlighted in triangles, and while the spray connected with flash are in circles. We found that there is a higher number of ACP when there is a reduced number of ACP when the sprays are coordinated with flash. If you think about the biology of the insect, they only lay the eggs on the bad breaks or unfair flash. It's normal to think that if we apply insecticide during that specific period of time, we will be able to reduce. But the problem that happened in Florida, that we have rain sometimes, and the plants just flash and grow faster. In the case of Texas, it's completely different because we are restricted to a certain period of time. But back to my point, we have the two different locations, and there is a reduced number of ACP when we coordinate the sprays with the phenology of the plant in both cases. Then on these trees, we determine the titer of silas. We use 8 leaves per trees, 10 trees per location. This is an example how the plants look when they are feeding. So for me to analyze, to determine the silas titer, I choose specifically those leaves, and then I remove I clean them, and I remove the central vein to to analyze the silas titer in them. So this is the result on them. In the location 1, there were a little bit differences, but the grower practice seems to have higher titer of silas compared with the spray coordinated with flash. In the location 2, that difference is completely lost. So and also this is telling us, it shows us that there is a temporal dynamic of the silas detection on mature leaves. So this is important it was important for us because using antibiotics or some bactericide, we can attack the bacteria when it's in the aerial part. That is part of the project, that part of the information that I left there, and is being studied by Dr. Kirsten Pell-Stalinsky and also Lucas Stalinsky. So this is just if you remember like how they look, this is a visual comparison between the number of ACP across over time with the silas titer, and we did not find any visual association. we analyze the correlation using Pearson correlations, and there is no association at all in any of the comparison that we did. So then I did like, but what is happening with the yield? Like, so I compared the 3 yield between the different spray practices. And what we found that when the grower apply pesticide by calendar instead of the spray connected with flash, those trees have a significantly reduced number of fruit per tree. So as soon as the tree is exposed to more insects, the yield is reduced. So that it means that there is a need for vector control. So no, we did not find any correlation between Silas titer and ACP density, which is interesting because I'm gonna show you what happened in the laboratory, in the microcosmos or the lab control conditions, and higher fruit yield were associated with lower SV population when spray were coordinated with flash cycle. And this is something that is being observed again during the last 2 years. So in the lab study, I evaluate the plant defenses against silas and herbivory to create tolerant varieties. That is the purpose of all the study that I did. Especially because plant response increasing the salicylic acid, which is involved in the plant defenses against pathogens, bacterial pathogens, and also to phloem feeders. So we evaluate the effect of silas inoculation frequency on HFV progression and plant defense response. The specific objective were the following: that we determine the silas titer in this group of plants, and
Freddie, your sound's gone.
Yeah, Freddie, we cannot hear you.
What you can do is like turn it off and turn it back on again. I still can't hear you.
oh, can you hear me now?
Yeah, now I can hear you. Perfect.
I'm sorry. I was talking about the specific objective, right?
Yeah.
I'm sorry. For a moment, this one get off. So we describe the salicylic acid induced plant defense response at varying silas inoculation frequencies. So the experimental design that I created was the following. The first one, I took 6 plants and we exposed them to the control that are uninfected ACP and silas, and the treatment that was the silas infected ACP. We exposed those plants for 7 days to this group of insects, to 20 insects per plant. That is the one that is going to tell us how the silas is going to move normally in a plant that is not exposed to any insects. Then we did the pulses inoculation, in which we repeat the same inoculation access period of 7 days each month, and we did it on both. And finally, is the continuous inoculation that you can think that probably that one is the one that can be considered the super infestation. And in here are the super infection, in which we compare both and see what happened. So we put insect on the plants and we keep them always, the insect was continuously reproducing on these plants. To analyze all the different assays I did, which I choose 12 mature leaves, and to determine exactly how I'm going to do this, I start to collect tissue from the tip of the leaf to the petiole, and I did it by month. So times 0, this is the first collection. 1 month later, the second, and then from now on until I reach 1 year of data. And this is what we found. we found that the bacteria, there is no differences between plants that were infected only were exposed to only one time to infected insects. So there is a certain amount of bacteria that the tree can harbor, and it was the same as the plant that were exposed continually to this insect. But what happened in here is that those trees that were exposed to continuously to this high pressure of insect, those plants die in the month 13. So I was losing a lot of I was losing those plants. So we did not find any differences between one time and continuous inoculation. What we did found is that those trees exposed to infected insects by pulses, those trees had the lowest amount of Silas titer, and this happened only during the first year, because then after the like 6 months later, after I finished the experiment, I went there to collect tissue of these plants and analyze the Silas titer, and in that case they reached the same level as the one-time inoculation. So there is an effect that is protective of the insect, like the insect can, because I forget to mention this part. I only use mature leaves in the case of the pulses inoculation. So the insect was exposed only to mature leaves. So if we use the same one over and over and over, it seems that the plant can respond. In the case of the one-time inoculation and pulses and continuous inoculation, the insect can reach all the tissues, the young and the mature. In the case of the pulses, they only reach the mature tissue. So we did not find any differences between one-time and continuous inoculation. And this one is telling us, are the pesticide application important if trees have the same Silas titer? Yes, it's important, because those trees that were exposed to a reproducing population of ACP killed the trees after 1 year. So then I was like studying, like, how can we manage this one in the field, and exactly what is I'm going to study. I'm studying the salicylic acid. This is a model of the salicylic acid metabolism and signaling in citrus. We have the following members in this model, like salicylic acid is synthesized by these two pathways. Then when we have salicylic acid in the cytosol, and it creates an oxidation, a reduction of the NPR that are in are polymers of NPR, and then is by using the thioredoxin is reduced to monomers of NPR. In that moment, the NPR can cross the nucleus or transport direct to the nucleus and activate the expression of PR1 as a negative regulator of this pathway are NPR3 and NPR4. And salicylic acid can also be transformed to different molecules that have been involved in response to pathogens, and also in the attraction to ACP and parasitic wasp. In the case of salicylic acid, can be transformed to the 2,3 and 2,5 dihydrobenzoic acid. In the case of the salicylic acid, the volatile molecule and also involved in the systemic acquired resistance in plant is the methyl salicylate that is synthesized by the VSMT, and then also the methyl salicylate can be transformed to salicylic acid. So we analyzed the expression of several genes. The first one was NPR1. And in here, like I have been observing during the years in one long being that they only do one time point or max two time points in the infection. Lately, is the time points have been increased. But if you remember that the bacteria has different point in which you can find those ones, so I associate the time that they found it with the vegetative growth. So in each of these colors is when the plant was growing and showing bud breaks and also growing shoots. So in here, like when they say one time, first D is like the first time that the bacteria was found in mature leaves, the second and the fourth time. And you can see how the expression changed just over time. Like that is the beauty of the studies when you have several time points, because you can compare for real how this gene move in the how this gene behave, the expression of the gene behave in the plant in different treatments like control and what we did. So technically we did find a higher expression on the infected ones. It's important to say that the white bars are the uninfected plants, or the plants that were exposed to uninfected insects, and the black bars are the plants that were exposed to infected ones. In the case of NPR3, this is what happened with NPR4, with a huge reduction of the expression on the second time. And that reduction in the expression of NPR4 was associated with a high expression of PR1 on those samples. It was like really interesting. Like if it was the real negative regulator, without the negative regulator, there is a higher expression of PR1. And also, I quantified the salicylic acid. I also quantified the methyl salicylate and the molecules that I showed you before, but I may not have time, enough time to reach all of that in this presentation. We found that the infected ones has a higher salicylic acid concentration on the leaves. And if you know, too much salicylic acid is detrimental to the plant, and those plants were stunted. That is exactly what happened. When there is a threshold of salicylic acid in which the plant can regulate the plant defense and grow, but if you surpass that threshold, those plants tend to be stunted and use all the energy into the immune defense. So this is important. We found the NPR4 was reduced. That reduction on the expression was associated with higher expression of PR1 and also with higher levels of salicylic acid. So this is the proposed model that we have. In one-time inoculation, the plants tend to respond, but if you add one factor, that is the more insects in there, I'm going to show you what is going to happen. So this is the continuous. There is over time, there is a reduction in the expression of NPR1, meaning that that plant is just collapsing. In the immune defense is collapsing. In the case of NPR3, we've observed the same, the same behavior, the same trend. In this, this is what we observe in the continuous inoculation. I'm sorry. So can you hear me again?
Yep.
Okay, so I highlight this one in here. And in the case of NPR3, there was a reduction of NPR3 in the continuous inoculation. So it seems that depending of what you have in the condition that we expose the plant, these one are going to respond differently. There is a reduction of NPR3 with certain amount, certain expression of PR and the PR1, and this one was associated with changes in the salicylic acid. And these are the too much salicylic acid for the plants, so those plants finally die. So this is the proposed model for the continuous inoculation, in which the high concentration of salicylic acid are detrimental, and it wasn't a proper response, a proper regulation of the gene associated with the local and systemic resistance. And when it was too low concentration of salicylic acid, we also cannot activate the expression of immune defense genes. In the pulses inoculation was a little bit similar to the continuous inoculation, but what was surprising in this case, it was that the amount of salicylic acid at the beginning of the experiment on those plants was super high. like not super high. It's a bad thing, but was a was a large amount of salicylic acid, and finally it get low at the end, and it's when we start to observe the silas on those leaves. So the proposed model is similar in this case, high concentration of salicylic acid and low concentration. So the conclusion of my study, I think I'm going to finish on time, is that there is no differences in silas titer between plant exposed to infected vector for one time, or as compared with the plant exposed to continuously breeding silas infected insects. The plants that were inoculated monthly for an entire year had the lowest titer of silas, that plant that were inoculated only one time, on those that were constantly challenged by infected ACP. The pulses of herbivory cause a boost on plant defense, but that is at the beginning. That was only during the first year of the study. The second part of the second part of this study, those plants show the same titer. The silas titer result observed in the field trial shows similarities to those observed. I didn't have time to show a paper that I have in the temporal dynamic of silas in the lab and in the growth chambers versus the field, but the probability to find silas on mature leaves was associated with moments in which the plants were showing vegetative growth. Also, higher fruit yield were associated with lower ACP population when sprays were coordinated with flushing cycles using the bad break phenology model. So in resume, even if the plants are infected, the vector management is still needed under the HLV stress that to reduce the adverse effect of ACP herbivory. So what we deliver in Florida, it was we deliver a new approach for the timing insecticide application, improve the ACP management that we call the bad break model. The results suggest that maintaining an ACP population below a threshold of 1 ACP per tap can improve the fruit yield. And also, we identify candidate genes to develop future transgenic varieties expressing potential tolerance to silas infection and or deterrence against Diaphorina citri. For example, if we can reduce the expression of VSMT specifically on the fair flash, the insect may not smell the molecule, they are not going to lay the eggs on those transgenic plants. So that is what we did. We did the information. We released the information to the plant geneticists and breeders to find to determine that if they want to do an up regulation and down regulation, or we can finally just knock down the genes and observe the phenotype. So I'm going to thank you. The acknowledgement are for the project that for the agencies that provide the funds. My principal investigators, Dr. Lucas Estelinski, Mamoudou Setamo, and Dr. Jean Albrigo that passed away the last year. Our collaborators in the studies in molecular biology and LCMS, Dr. Kirsten Estelinski, Dr. Wang and June, and my friends and colleagues that I work in Florida, and of course, what I did also in Texas A&M, my advisor, Cecilia Tamborindegui, and my friends. And you see here, this is Chloe in her best time. And now I'm open for questions. Thank you very much.
Thank you, Freddie. Thank you for the excellent presentation. Any questions for Freddie?
You can also write in the chat if you want.
Great. Yes. So Freddie, I want to ask you a question about the citrus greening disease. You said the threshold is one psyllid per trap. How do you? So how many? Say how you place the trap? Say how many traps you place per tree?
3 taps. We do 3 taps in the southern and 3 taps in the northern side of the trees.
Okay, that's 1 per trap or per day.
Yes, 1 tap. Yeah.
Okay.
But if the tabs is lower than 1, is still is better. So the yield just increase more, are larger.
How often the growers have to spray the tree?
They in Florida and in here they spray 6 to 12 times depending of the ACP infestation. In Florida, they those growers were applying 8 times.
Wow.
So that is a lot of pesticide. That is, and in Florida, you will see that our lab has, like the Lucas and Zelinsky lab, have studies there that Dr. Chen is doing, in which the insects are already resistant to several insecticides, especially in the central Florida. So what we were thinking there is what they were doing, and also I was collaborating in that one, was the insecticide rotation and related with the model that we have. So we found differences between Movento and Exidel, and we using Exidel in the exact time can knock down the population of ACP almost for 2 months. In the case of Movento, that doesn't happen. It knocked down for 2 weeks, and then the population starts to grow up again.
Okay, thanks. Any other people have questions?
Hi, I have a quick clarification. So the CLAS bacteria, does its titer like just naturally fluctuate over the course of infection?
Yes.
But in the plant.
Yes, because the bacteria, what I didn't have enough time to mention this part, but the bacteria follow the nutrient. So if you know about the growing of citrus, they follow like vegetative grow, root grow, vegetative grow, root grow, vegetative grow, and root grow. And that happened over the whole season. So the bacteria is highly, the probability is high to find the bacteria when the plant is growing, the vegetative grow. Then when it change to the root grow, the titer goes down and all the bacteria is moving to the roots. And then from the roots, move to the aerial part and now on. And when that is happening, what the plant is doing is closing the phloem, closing the sieve elements, because the plant can respond to CLAS. It doesn't escape. But the response of that is closing the phloem. So when you close the phloem, what happens is there is no more movement of glucose. There is no more movement of nutrient. So finally, those branches die, and that is what happened. The dieback phenotype in citrus is exactly that. There is no more connection between the roots and the leaves, or if you want to say the roots, the other part, and the underground part of the tree. So the plant responds, but the bacteria just follow the nutrients.
Oh, okay. Do you ever test the roots themselves for titers? I guess.
Yeah.
Well,
There is a paper that was just published by Nehela and Dr. Chilini in University of Florida, in which they showed the model, and they found more bacteria in roots, specifically on those time points. Because I did not want to touch my roots. Because if I touch my root in my study, I'm going to completely change the aerial parts. I tried the first time and said, no, that is not a good idea.
Yeah, I imagine that'd be like hard to do without affecting like the plant's immune response.
Yeah, you change the plant immune response, and if you remove the roots, the plant is going to be weaker. So it's like, okay, no, I'm not going to do that. I'm going to just analyze the mature leaves. So you analyze what you can.
Thank you.
Thank you for your question.
I have a follow-up for that. Why did you choose the mature leaves again and not necessarily the immatures?
Because what I did not choose the immature, it was because we want to know exactly when we find the silas on those leaves. And the immature leaves has the problem that they harbor nymphs. So when to compare against all of them, all of my treatment, there is no purpose, because if in one time inoculation I will be able to collect those leaves, but from the continuous inoculation, I will not be able to do it because they have names on them. That is the reason. Like it was a technical reason that we did not choose new shoots or new vegetative growth to do the analysis.
Okay, thank you.
You're welcome.
I have a question. So of all the pest species that you were interacting with in Weslaco, what are the primary pests you'll be working with? What's the future for Freddie?
The future for me? Well, right now I'm studying the insecticide resistance of whiteflies. Also studying the spot cucumber beetle, preference on different varieties of watermelon and potato psyllid, and probably the Asian citrus psyllid, but in collaboration with Dr. Kranti Mandadi in this center, and also Veronica Ancona and Mahmoud Setamu in the Citrus Research Center. And also the Mexican rice borer, there is a problem here too. So they like at least 5.
I have a follow-up question about the citrus greening disease. So what's the current status in the US? How it is still spreading, and how people would prevent the spread or reduce the prevalence?
Well, the only way to reduce the spread of this disease is eliminating, eradicating the Asian citrus psyllid. The problem is that we cannot eradicate it from Florida. We have tried to reduce the population, but the eradication has not been achieved. And right now, what happened in Texas, if you know, we have a huge freezing, a huge winter storm that just decimated our citrus growths. They just die, the grapefruit in here. So as soon as you don't get the connections, if you think about US or the southern part, going from California to Florida, we have citrus in several areas, and there are citrus that are not infected, especially in Arizona, but they are too small areas that has. But in California, the disease is spreading, but they have a more drastic way to control the spreading of silas, in which as soon as they detect the bacteria there, they remove those trees.
Okay, wow.
And that is the only way. What happened in Florida is a completely special case, and it's really hard to explain because the growers just abandon those trees. They abandon the groves. They don't, okay, I don't want to manage it anymore. So they just abandon and those trees keep surviving for 2 or 3 years after they abandon those groves. So the insects are just thriving in there, and that is a niche. And the problem with Florida, we don't have a centralized control. There is an agency that is centralized to create strategies to reduce the ACP, but it's not as powerful as the growers. So if we want to eliminate for real ACP, all of us need to follow the strategies that we recommend, and from there start. But what happened is you have organic growers that doesn't want to apply anything, and the commercial one that apply pesticide 8 to 12 times in a year. So it is a miscommunication, if you want to say, like if we haven't reached properly the audience.
Okay.
So that is the thing, that to do this one, even to do it in here, because in Texas, in Weslaco specifically, there is a huge problem with the whiteflies. Why there is a huge problem with the whitefly is because the cotton that is planted nearby do the defoliation in August or September. Around that time, they do the defoliation. But if you do the defoliation without applying pesticide or without controlling your population, all those whiteflies move from the cotton fields to the vegetables and cruciferous crops. And those are being attacked by whiteflies. And what happened in the vegetable crops is the same thing. After they do the harvest, they just abandon those crops and keep it there for 1 month or 2 months after harvesting, which is bad too, because to eradicate or to reduce the population of pests, we need to control them in the proper time. So I'm writing right now a grant in which I'm proposing a model in which we are going to apply a pesticide before defoliation and after harvest in vegetable crops to see what happened with the whitefly population in the Rio Grande Valley.
Okay, I guess we can stop now because it's already 12:03. For those registered for the class, we can continue the discussion for another hour. Others can leave. Thank you, Freddie, for the excellent seminar.
Thank you so much for inviting me. Thank you, Chloe.
And we can have a 5 minutes break and come back at maybe 12:10.
Okay.
Okay, all right.
Awesome.
Okay.
Thank you.
Mhm.